My name is Elton Zekiraj and I'm a group leader here at the Astbury Centre.
My name is Miriam Walden and I'm a postdoctoral scientist in the lab of Dr. Elton Zekiraj. We are studying cell signalling via the small protein ubiquitin.
Typically, this small protein acts as a tag for other proteins to be sent around the cell or to be recycled by the cell.
We specifically studied ubiquitin signaling in the immune response. So as an immune signal comes into the cell, there's a protein known as BRISC and SHMT2 that are involved in transmitting that signal to the nucleus of the cell and allowing that signal to continue in order to fight off whatever it is that is triggering the immune response.
Typically in our laboratory we use cryo-electron microscopy to identify that three-dimensional structure of proteins. And that often teaches us about how these proteins function. We also use protein engineering and protein production using viral infection and insect cells. We then characterize them using biophysical and biochemical methods to determine exactly what they do. In this particular paper, we have discovered how BRISC-SHMT2 regulates the turnover of interferon receptors.
When we take a look at BRISC, we can see it's actually a dimeric structure in itself, made up of 4 different subunits. You have the arms here, which consist of MERIT40 and BRCC45, and then you have a core here that consists of the actual active enzyme BRCC36 and Abraxas2. And this is the actual enzyme that will perform the deubiquitylation. What we discovered in the paper is that it actually also interacts with a 5th enzyme known as SHMT2. And here, this is the SHMT2 protein. And this protein is usually involved in DNA metabolism. When vitamin B6 binds, another version of the SHMT2 comes along, and you get this tetrameric structure. And this is an active enzyme. We've discovered actually that the dimeric protein is capable of coming along and binding to the BRISC. And as I hope you can see, the tetrameric version would not be able to bind in the same space.
We realized that only the dimer in the absence of vitamin B6 can bind BRISC and can function in cell signaling or interference signaling. We have figured out a really unexpected cell signaling pathway that has taught us about how we fight infections.